gives rise to phenotypic variation, and of course this is important for understanding the genetics of diseases as well as understanding genetic variation in evolution. And here's a reproduction of a classic photograph showing variation in height in a set of college students ranging from 5 feet tall to 6.5 feet tall, and the men are in dark and the women are in white-colored clothing. And it's been known for a long time that height is an extremely heritable trait. So here's a classic plot that was made by Francis Galton, and this that's showing the on the x-axis the average height of parents, on the y-axis the average height of offspring. He shows that they're extremely correlated, and this gives rise to the notion of regression. And in the last, so it's been known since Galton, at least, that and probably anecdotally longer, that height's a very heritable trait. And in the last 5 or 8 years or so, there's been a lot of progress actually in starting to dissect the genetic basis of traits like height. Here's a relatively old plot now from a paper from 2008, but I think it's quite a nice plot, which shows so in this study, they identified 20 different loci, where for each of these loci there's a SNP that influences height by on the order of 3 to 5 millimeters each, and so you can classify the alleles in each individual as the alleles at each site as being a tall allele or a short allele, and if you sum up the number of tall alleles that each individual has, you can predict to some extent what their height is. So using 20 loci, the range between the top predictions and the bottom predictions is about 6 centimeters. There are now on the order of several hundred height loci that have been identified. So one of the key questions then is to understand a bit more about how loci, how variants like this actually affect phenotypes. And at a very basic level, of course, the genome, you can think of the genome as doing, sort of in a very cartoonish way, it's doing two sorts of things. So one kind of thing that the genome does is it encodes protein coding genes. So part of the DNA sequence here serves as a template for making transcripts. This is messenger RNA, and then the messenger RNA is translated into protein. And so a couple percent of the genome is devoted to encoding this genetic information, the protein coding gene information. And then there's also information in the genome that tells the cell when to make each gene and how much to make, and of course different cell types are making different proteins. And so if you think about genetic variation and how it impacts phenotypes, you can think of it doing this very broadly speaking in two kinds of ways. You can imagine genetic variants that are affecting the sequence that encodes proteins, as well as information that's changing gene regulation. So it's changing how much of any given protein you're going to make and under what circumstances and so on. And we know we've known for a long time quite how to annotate protein coding regions, and protein coding annotations now are very accurate. But it's turned out to be a much more challenging problem to identify regulatory sequences and to understand exactly how they work. However, it is now becoming clear that regulatory variants play a really central role in complex traits. And here's one example plot. There are many examples that one could have used here. So the x-axis shows position across a particular region, and then each dot here shows the level of association between a SNP and Crohn's disease. And down below you can see the locations of genes, and so there's a region here where there are a bunch of SNPs that are all statistically associated with risk of Crohn's disease. Presumably there's one site in here which is causal, and then a bunch of other variants that are in linkage disequilibrium with a causal site. And this this variant apparently lies outside of any protein coding region. And so we might infer from this that it's most likely doing something regulatory. But it's a great deal of work at present to go from a signal like this to figuring out which is actually the causal site, what does it do, what's the relevant cell type, which gene is it affecting, and so on. And it turns out that this is actually this kind of example is very typical. So here's a figure from a very nice paper from Joe Pickrell, which is posted in bioRxiv at the end of last year, where he estimates the proportion of hits in genome-wide association studies that can be explained by non-synonymous sites, i.e. sites that are changing protein coding sequences. And you can see for a bunch of different traits that the estimates are ranging from something like 2 or 3% up to about 18 or 20%. So for all of the traits that he looked at, the vast majority of the associated SNPs are non-coding, and so in general we don't really understand the mechanisms of these, but the natural inference is that they're affecting gene regulation in one way or another. So let's just think about how this might work. So from molecular biology, we know quite a lot of the key players in gene regulation. There are probably more to be found, but we know many different players. But we still don't really know how to interpret the regulatory information that's encoded in DNA sequences. So if you think about the process of transcription, so here on this plot, this gray bit's supposed to represent the start of a coding region. To make a protein, you have to start by transcribing messenger RNA. So this RNA polymerase is the machine that does that, and then you need to put together a lot of other machinery in order to get the polymerase properly assembled and get transcription going. And so this machinery relies on DNA sequences that encode specific information, both close to the transcription start site as well as at more distant locations that are often called, many of these are called enhancers. And so presumably, variants that are affecting complex traits, but not directly through proteins, in many cases they'll be affecting the function of these kinds of regulatory elements. But it's still, as I said, at this point it's very difficult to look at the DNA sequence and actually figure out what any bit of sequence does. And so this is a problem that we and many other groups in the field have been trying to tackle. We'd like to understand first what are the primary kinds of mechanisms by which SNPs impact gene regulation, and ultimately we'd like to get much better at interpreting non-coding variants that affect phenotypes to understand, to be able to figure out for a particular variant what's the relevant cell type in which it's affecting regulation, which genes is it likely to be affecting, and so on. So to tackle this, the major approach that we have taken is using an approach called expression quantitative trait locus mapping. So we use this as a tool for linking genetic variation to changes in gene regulation. So for those of you who are not familiar with this, let me just talk you through it quickly. So the basic idea is that we'll collect a particular cell type or tissue type from a whole panel of individuals. So in the studies I'll describe, it's about 70 individuals. And then we'll use a genome-wide technology like expression microarrays or RNA sequencing to measure gene expression for essentially all of the genes in the genome. And so now, for each individual and each gene, we've got an expression measurement, and then we can ask whether there are variant SNPs in the neighborhood of each gene that are affecting gene expression. So here, in this case, perhaps the white-colored allele leads to lower expression output than the green-colored allele does here. And so what we'd like to be able to do is to identify variants like this that have effects on expression of nearby genes, and then to understand why they do this. What are the kinds of mechanisms that cause this? And so I should mention that in this talk I'm going to be focusing on what are referred to as cis-EQTLs. This means operationally, this means EQTLs where the SNP is close to the target gene. There are also trans-EQTLs where you've got a SNP somewhere else in the genome, and I won't talk about those because well, the mechanisms of trans-EQTLs are very different from the mechanisms of cis-EQTLs, and they're also more difficult to detect. So let me just give you one example of an EQTL. This is for the HLA-C gene, and so these are data from microarrays. Each dot shows you the expression level of HLA-C for a single individual, stratified by genotype here. So you can see that if you've got the CC genotype, you're expressing this gene at a higher average level than individuals with the AA genotype. And it turns out that this EQTL is also associated with HIV-1 progression through its effect on HLA-C levels. So we decided to use a set of cell lines known as the HapMap cell lines as a kind of model system for studying expression variation. And this is all joint work with my collaborator at the University of Chicago, Yoav Gilad. So we've been working with a panel of about 75 cell lines from the Yoruba population in Nigeria. And so these are cell lines that were created by transforming B cells, which are a component of the immune system, into an immortalized resource that you can grow. And originally the idea with these cell lines was that there'd be a renewable resource for DNA for the HapMap project and more recently the 1,000 Genomes project. But a number of people, including Vivian Chung and others, realized that you can actually use these cell lines as a resource for studying cellular phenotypes. And so we've taken this approach where we take these cell lines that have been very thoroughly characterized by the 1,000 Genomes project. We've got essentially a complete catalog of genetic variation in all of these cell lines. There's also a lot of functional information from one of these cell lines that's been collected by the ENCODE project. And so we've been using this as a kind of model system for understanding these links between genetic variation and regulation. So let me just give you an example of what one of these cis-EQTLs looks like. So these are data from an RNA sequencing experiment that we did a few years ago. The x-axis here represents position along the genome. The y-axis represents read depth of RNA sequencing data. So this is essentially a measure of the abundance of messenger RNA transcript from this region. And the three lines here show the data stratified according to genotype at a particular SNP in this region. So if you've got the GG genotype, then you've got this gene turned on. If you've got the AA genotype, then this gene is basically turned off. So we'd like to understand more about how a SNP like this, or a SNP that it's in LD with, actually acts. So from that study, we identified about 1,000 cis-EQTLs. So one of the first kinds of questions you might ask is just sort of very basic descriptive questions. So what's the context of the variants that cause EQTLs? First of all, where are they with respect to the genes that they regulate? Now, it turns out this isn't a totally straightforward question to answer because of linkage disequilibrium. So linkage disequilibrium refers to the property that SNPs that are close together in the genome are very often correlated in their genotypes. And so here's an example where each dot shows you the p-value of association between a SNP in the region of this gene SLC12A7 and an expression of this. And you can see that there are a bunch of SNPs that are all very significant. Presumably one of these SNPs is a causal site, and then the others are simply in linkage disequilibrium with that causal site. Now I would note that here we're using 1,000 genomes data, so we've got representation of nearly all of the common SNPs in this region, or I should say nearly all of the SNPs that are in the positions that are variable in the sample. So we can be fairly confident that we've actually got the causal site here, but it's still ambiguous about which site actually is causal. So with this type of data, it's difficult to answer a seemingly simple question like where do causal sites lie around the target genes? And so to tackle this, a few years ago we developed a Bayesian hierarchical model. And the basic idea for this is that we've got analysis at two levels. So there's analysis at the level of genes, So at the level of genes, we can measure the strength of association between SNPs and gene expression. So here these the strength of association is measured as Bayes factors. And each SNP is going to be annotated according to various kinds of putative regulatory annotations or distance from genes, location with respect to genes, and so on. And then at the global level, we can use all the genes in the genome to ask about the relative enrichment of causal sites in each type of annotation. And so the idea is that we parameterize a prior probability that any given SNP will be a causal EQTL for each gene as a logistic function of the annotations that it sits in with a prior parameter for each annotation, and then we're going to use all of the genes in the genome to estimate these prior parameters. And so you can imagine fitting this as sort of an EM algorithm where you're using all of the genes in all of the QTLs in the genome to learn about these prior parameters, and then these can in turn reweight the posterior probabilities in each region. And so you can get a maximum likelihood estimate of the enrichments for every category. And so one really nice feature of this type of model is that it gives us a principled way of estimating the relative enrichments the relative importance of different kinds of regulatory features in a complicated model where we've got a bunch of different features simultaneously. And it's actually quite difficult to do this in a really principled way using sort of naive methods of just sort of measuring enrichment. So just to give you an example of this, here's a plot that shows you basically a density plot of the locations of EQTLs around target genes. So in the top plot, we're just taking the most significant SNP for each EQTL. So basically, if there's a SNP that's significant above some threshold, then we plot the location of the most significant SNP for that gene. So this shows you the upstream region for each gene. The black bit shows you the transcript, and the right-hand side shows you the downstream region. And this is our estimated density that we get from the hierarchical model. And you can see that this strongly downweights these long-range effects, and it concludes that this peak of EQTLs in the vicinity of the transcription start site is much stronger. Yes?
What's on the left-hand side? What's an EQTN?
Yes, apologies. So EQTN here, the N is referring to nucleotides, so this is an estimate of the density of causal sites. So, sorry, I skipped that. But so basically, the distinction is that in terminology is that an EQTL is a location where we think that there's a variant that's associated with the expression, but it may be ambiguous which is the causal site. And then the EQTN is our estimate of which site is causal. Yeah, thank you for the question. So anyway, some features that you see from this are, first of all, there's a very strong concentration of EQTLs around, or EQTNs around transcription start sites, which makes a lot of sense because a lot of the regulatory machinery is there. There's generally enrichment throughout the transcribed region. So it seems like the transcribed region sort of sets up a domain of regulation. So for very long genes, it's more likely that you've got regulatory regions at a distance downstream from the transcription start site than in short genes. And also, we find that there are relatively few really long-range EQTLs, although they do exist. But they're a small minority of all EQTLs. Okay. So to summarize quite a lot of other work that we've done, we found that most EQTLs lie inside or near to their target genes. We also find that they're enriched in active chromatin regions and in transcription factor binding motifs. But in some ways, just doing computational analysis of the the EQTLs was a bit unsatisfying because we found it was really hard to make sense of the vast majority of EQTLs. And so the next question was how to dig deeper into these mechanisms. And so to tackle this problem, we thought about the mechanisms of EQTLs in the following way. So when you have an EQTL, basically what you observe is that there's an association between a SNP and the steady state levels of mature mRNA in the cell. So what are the kinds of processes that determine these steady state levels? Well, you've got sort of a life cycle of mRNA that starts with epigenetic modifications and priming the chromatin through transcription, processing of the pre-mRNA, and also at the end of the life of mRNA, there are processes like RNA decay and RNA degradation that are removing mRNA from the system. So the amount of mRNA at steady state represents a balance between these kinds of processes and removal of mRNA from the system. I'm not going to talk about it today, but of course there are also very important steps in gene regulation at the stages of translation and the controls of mature protein levels. And we're starting to do a bunch of work in this area as well, which shows that actually there is a lot of cis-acting variation affecting translation and perhaps mature protein levels as well. But we're not as far along in that work, and we don't understand that as well. So to make sense of, so to learn more about these EQTL SNPs, our idea was that we can take a variety of different genome-wide assays that have been developed in the last few years for querying each of these different stages of gene regulation. And then if an EQTL acts through altering any one of these features, so for example, by altering histone marks or chromatin accessibility, then this EQTL SNP should also be a QTL for these genomic assays, and then you would see that there's an effect that gets carried on through all the stages of the mRNA life cycle from that point onwards. And so today I'm going to tell you just about work that we've done at this first step of chromatin priming, because we think this is a particularly important stage at which EQTLs are most often acting. So I'm going to start with telling you a little bit about work that we've done with DNase-1. And so DNase-1 is an enzyme that's been used for a long time, since the 70s, as a tool for identifying regions of active chromatin. So if you think about the DNA in the cell, most of the DNA is wrapped up around nucleosomes, and so it's tightly packaged. It's relatively inaccessible to gene regulation. So an important step of gene regulation is that the DNA is opening up, nucleosomes are getting ejected, and transcription factors are coming in and binding the DNA and enacting programs of gene regulation. And so when you have active gene regulation, usually you get regions regions where the DNA is relatively open and unprotected by proteins like nucleosomes. And so DNase-1 is an enzyme that can come in and clip the DNA sequence in these open regions. And so basically the assay is that you treat the cells with this DNase-1 enzyme, you get these cut points, and then you take the fragments and sequence from the cut points on next generation sequencer, and you get a profile of basically the sensitivity of each location in the genome to cutting by DNase-1. And here's an example of what the DNase-1 profile looks like in average around the binding site of a particular transcription factor called NRSF. So NRSF has a binding site of about 20 base pairs where it's sitting on the DNA sequence, and the actual location where NRSF is sitting protects the DNA from cutting by DNase-1. But then off on the left and the right, you've got high rates of cutting because the DNA is relatively open. And then as you go further off in either direction, so scale here's about 50 base pairs, you're moving into nucleosomal regions where the DNA is protected. So the idea is that DNase-1 gives us a relatively generic assay of active regulatory regions, and We've also done quite a bit of work on using this kind of signal to actually identify transcription factor binding sites more generally for a very large number of transcription factors. I should mention that other groups like Greg Crawford's group and John Stamatoianopoulos' lab have also done a lot of work with DNase I and also developing methods in this area. So the idea that we had was that since DNase I is providing this information about active chromatin for a large variety of types of active chromatin, we had the idea that by looking at variation in DNase I sensitivity across individuals, we'd be able to identify SNPs that are affecting this early stage of gene regulation. And so we did DNase I sequencing in 70 Yoruba cell lines, which was quite a challenging experiment, which was led by Afma Pai, who is a wonderful graduate student in Yoav Gilad's lab. So the basic idea here then is that we've got 70 individuals. We get DNase I sequencing data for each individual, so these are short reads, map them onto the genome, identify regions that are sensitive to DNase I cleavage in at least a subset of the individuals, so these are often referred to as DNase I hypersensitive sites. And then we look for genetic variants that are associated with sensitivity here. So perhaps the pink allele here is associated with higher sensitivity. And so we refer to these as dsQTLs, so DNase I sensitivity QTLs. And so when we did this experiment, we found that actually there's a tremendous amount of variation in the genome in DNase I sensitivity. So we identified more than 8,000 dsQTLs across the genome. And here are what the QQ plots look like. So for this, we're taking two different window sizes around each DNase hypersensitive site. So the green dots are taking a 2 kb region window centered on each hypersensitive site and then testing every SNP in that window for association with the hypersensitive site. The black dots are for a 40 kb wide window. And you get the sense from these QQ plots that, first of all, there's a tremendous amount of dsQTL variation. And then secondly, that most of the regulation of dsQTLs is over very short distances. And I won't show you the hierarchical model results, but they're very consistent with this intuitive notion you're getting from this. So let me just show you an example of what these things look like. So this is about a 200 base pair region, and the three lines here show you the DNA sensitivity of this region stratified by the genotype at a SNP that's right in the center of the region. So if you've got the CC genotype, then the sensitivity of this region is about 10-fold the genome-wide average. If you've got the TT genotype, then this region's basically closed off, so the sensitivity here looks like the background signal. It turns out that this particular dsQTL seems to be due to disruption of a binding site of a transcription factor called NF-kappa B. So there's a match to the NF-kappa B motif at this particular location, and the C allele here matches the preferred binding site for NF-kappa B. The T completely wipes out that binding site. And so our inference is that if you destroy that binding site, then NF-kappa B can't get in here, and so then the whole region gets covered over in nucleosomes, and it becomes insensitive to DNase I. We can get some evidence for the presence of NF-kappa B from independent data from Mike Snyder's lab. So these are ChIP-seq data for NF-kappa B, also in lymphoblastoid cell lines, and you can see that there's a peak of NF-kappa B binding here for the CC genotype, but there's no NF-kappa B binding in TT individuals. So the next kind of question is, do these dsQTLs that we've identified affect gene expression? And happily the answer is that they do. So here's one example. Here's a dsQTL. So there's a SNP in the middle of this sensitive region. It's a couple hundred base pairs wide. So the AA genotype is relatively sensitive. The GG genotype is relatively insensitive. And this gene, this hypersensitive site, is in the neighborhood of this gene here. And you can see that the sensitive genotype for this DHS located here produces relatively high gene expression. The GG genotype has this gene virtually turned off. And we find this is a very common pattern. So we estimate that using our current data, about 55% of EQTLs are associated with changes in DNase1 sensitivity, and there are some reasons why there are some dsQTLs that we miss, so the actual answer may even be higher than this. So this seems to be one fundamental mechanism by which dsQTLs are generated. Yes?
They always show these almost perfect codominance patterns.
You mean that the heterozygote is intermediate? Yes. So generally we think that these act, that the two chromosome copies act independently through a sort of a cis-acting mechanism, and so I don't think we've found any good examples of departures from additivity. I mean, there may be some that we haven't found yet. So anyway, so here's a QQ plot where what we're doing here is we're taking dsQTLs and then we're asking whether they're associated with expression of nearby genes, and you can see there's a tremendous amount of signal here. So it's a two-sided QQ plot, because you can imagine on the right-hand side you can have an increase in DNA. The allele that increases DNase1 sensitivity is associated with increases in expression, and on the left-hand side you can have increases in sensitivity go along with decreased expression. So you can see that there are more of these activating dsQTLs than there are repressing dsQTLs, and this is about a factor of 2 to 1. You can also see that for dsQTLs that are very close to the transcription start site, quite a large fraction of these are associated with changes in expression, although there are also longer range effects. So we next wanted to understand a bit more about how dsQTLs are changing chromatin function. And so to do this, we collected additional data on a variety of different assays that are querying different aspects of gene regulation. So we looked at nucleosome positioning, a bunch of histone marks, RNA polymerase II, DNA methylation. And what we find is that these dsQTL SNPs are typically driving very coordinated changes in a whole suite of different aspects of chromatin. So these different aspects of chromatin are really co-varying together, and individual SNPs are able to toggle the levels of all of these things simultaneously. So here's one example where the different tracks here are showing the results of different assays, and the different colors here show you the read depths segregated by genotype. So the dark blue is AA, and there's AG and GG. And so you can see that the AA individuals have got high DNase1 sensitivity, they've got high RNA polymerase, and they've got high marking by 2 different histone marks that are associated with active chromatin. And finally, they've got high RNA-seq levels. So this shows you expression of the first exon of this gene. And so you can see in this particular example that one SNP is producing very coordinated changes in basically everything that we looked at. And this is a relatively clean example, but in general we estimate that the correlations between these different molecular factors are very high. We also find the data here are a bit noisy, but we find that there's generally quite a strong correlation between DNase1 sensitivity on the x-axis and transcription factor binding data from ENCODE. The data are noisy here because we're only using one individual, and there's not very good read depth. Secondly, we find that DNase1 sensitivity is associated with systematic changes in nucleosome positioning around the affected sites. So this shows the average density of nucleosomes centered on DNase1 QTLs, and here that the different lines are averaging the high heterozygote and low DNase1 genotypes. So you can see that the genotypes that are associated with high DNase1 sensitivity, I showed you they're also associated with high transcription factor occupancy. They tend to have low nucleosome occupancy at the center of the dsQTL, and then very strong nucleosome positioning compared to the other genotypes. Now, one thing I would note here is that even in these active regulatory regions, there's still substantial nucleosome occupancy. And the way to think about this is that all of these assays that we do are on a population of cells. And so at any given time, an active regulatory region, we believe, is only actually active in a modest fraction of the cells in the population. And it's probably a dynamic process, although it's still very difficult to measure the dynamics. I'll come back to this later, but what we think is that these dsQTLs are probably shifting the fraction of cells that are active in any given region from, let's say, 30% to 20%. They tend not to be binary changes between on and off states. We also find that there are sites that create long-distance changes in chromatin. So these will be examples. So these will be dsQTLs that are affecting expression at distant promoters. So here's one example. So these are tracks for a bunch of different chromatin features. Here's an enhancer region. There's a SNP in this enhancer that affects DNase 1 sensitivity and histone marking here. It also affects the function of this promoter, which is about 25 kilobases away. And this is quite a common kind of feature, that you have long-range effects on chromatin from one site to a distant promoter. So in the last part of the talk, I just want to say a little bit about trying to understand the precise sequence features that are being impacted by these dsQTLs. And so I think that one really nice aspect of studying genetic variation and gene regulation is that we can start to get at the causal pathways between DNA sequence and gene regulation. So one real challenge in the genomics field is that we can measure many different things, and we find that you get strong correlations between many different features that you measure, but it's generally been quite difficult to figure out what things are causally upstream of what. So for example, it's been shown that histone marks are highly correlated with one another and also are able to predict transcription factor binding and active enhancers and promoters and so on. And so a lot of the language in the field has suggested that perhaps these histone marks are causally upstream of the function of the enhancers. But it's difficult to tell from the genomic data alone what's the order of the mechanism. But of course we know that DNA sequence variation is extremely stable. So genotype is extremely stable. So that must be causally upstream of any of the genomic assays that we measure. So the punchline is going to be that we find that in many cases these chromatin QTLs act by disrupting transcription factor binding sites, and that these changes in transcription factor binding can play causal roles in altering everything else that we've measured. So let me just talk you through the logic of this in a cartoon. So here, let's suppose that we focus on a particular transcription factor, like let's say PU1, and we can identify all of the, so we've got estimates of where PU1 is binding across the genome, and then we can identify all of the SNPs that sit in PU1 binding motifs. And now, for each of those cases where we've got a SNP and a PU1 binding site, we can classify the two alleles according to which allele is predicted to have better PU1 binding and which is predicted to have worse PU1 binding. So here the X is supposed to indicate the allele that is predicted to have worse PU1 binding. And now we can ask whether the direction of effect here on the transcription factor is associated with aggregate changes in any of the other kinds of features that we've measured. So under the null hypothesis, we might expect, if we look, for example, at histone marking in the neighborhood of these transcription factors, if binding of the transcription factor is not causal of histone marking, then we should see that if there's any change at all in histone marking across genotypes, the direction should be random. So on average, there should be a zero effect on histone marking. Conversely, if binding of the transcription factor causes, for example, an increase in marking by a, quote, active histone mark, then we should see on average that the allele that is predicted of better binding should be associated with increases in histone marking. And in fact, we see this very systematically. So here are the data for PU1. These show you the fitted regression lines for a bunch of the different for different things that we measured. So let me just explain this. The x-axis here takes the two alleles in a SNP, and it measures the difference in predicted transcription factor binding between the genome reference allele and the alternate allele. So the right-hand side means that the reference allele has better binding than better predicted binding than the alternate allele of the SNP. And then the y-axis shows the mean difference in ChIP-seq read depth in heterozygotes. And so what you can see is that there are all of these factors here that are associated with active chromatin are systematically increasing when on the allele that we predict should have better binding for PU1. And conversely, K27 trimethylation, which is a mark that's associated with repression of chromatin, has the reverse direction. More globally, when we look across all transcription factors, we find that in aggregate, there's an effect that globally all of the transcription factors are associated with increases in all of the active histone marks in POL2. And here are a bunch of the particular transcription factors that are most significant. There's one exception that we identified in the data, which is NRSF. And NRSF is a well-known repressive factor that's associated with the polycomb repressive complex, and polycomb repressive complex is important for laying down this K27 trimethylation, and you can see that this has got a very different pattern from all of the other transcription factors for which we found signals in these data. So just to summarize this last part of the talk, what we think is that most of the regulatory regions have got competition between these active and inactive states, these active and inactive chromatin configurations. And so there'll be this active configuration where there's transcription factor binding and high DNase 1 sensitivity and strong nucleosome positioning and histone marking and so on. And then this inactive state where the regions are basically closed down. And we don't have direct measurements of this, but my guess is that this is probably a relatively dynamic process where the chromosomes are flipping between the two states. And then the idea is that if you've got SNPs that weaken transcription factor binding, then that will tend to push the system in this direction. And as I said, most of the SNPs that are common variation in the population, which are the things that we can actually detect as QTLs, tend to have relatively weak effects. So they presumably weaken the ability of a TF to bind in a particular location, and that will tend to shift the equilibrium in this direction, but only in a quantitative way. And then a fraction of these dsQTLs then go on and affect gene expression. Okay, so just to summarize, so we find that that a major set of cis EQTLs act through these changes in chromatin that are sort of very highly coordinated changes in multiple aspects of chromatin in a quantitative way. And I didn't have time to talk about it today, but we've got some new data on studying QTLs that affect translation and steady-state protein levels, and I think this is going to be a very interesting story as well. So this is something that the mRNA QTLs can't tell us about at all. So let me just thank the key people who have been involved in this work. So Yoav is my experimental collaborator in Chicago. Matthew Stevens has been involved with quite a lot of the statistical work. Athma is a really key experimental person in Yoav's lab who helped to spearhead a lot of the early work that we did in this area. Jack and Roger led the DNase1 work, and Graham and Bryce did all of the histone work, and Alexis is involved in the project now to sort of put together all the things that we've measured so far, as well as the protein stuff that I alluded to. So I'll end there.
Any questions for Jonathan?
Mark.
Jonathan,
Yes.
did you miss something? Did you say something about population level variation?
Do you mean between populations?
Yes.
So I didn't talk about it. So there's been some work on that, although we haven't contributed much of it. So for EQTLs there's some debate about whether there are good examples of population-specific EQTLs. So there's sort of the trivial case where you've got a SNP that's an EQTL that's not segregating in different populations, but it's not really clear whether there are interesting population-specific EQTLs where you have a variant that's segregating in two populations, but there's only an EQTL in one of them. I don't think that there are good examples of that people believe. There's been a bit of work on, so Mike Snyder's lab has done some of this work on population-specific variation in chromatin, and I think again probably the vast majority of that is driven by differences in SNP allele frequencies, but it's certainly present.
Hunter has shown quite strong differences between Yoruba and European on the potential.
That's the DNA methylation, right?
Yeah.
Yes.
methylation.
Yes. Yeah. Thank you. Yeah. Yeah.
So in this talk, you've looked at sort of SNPs affecting mean expression.
Yes.
Have you looked at higher moments like variance, for example? I mean, you need bigger, larger sample sizes for that. But do you think it would be interesting to look at variance in gene expression?
Good question. The question is about variance in gene expression. So we haven't done any of that. I'm just trying to think. I think there's been some EQTL work on variance. So we haven't done any of that. I think that, for studying variance, you have to be a little bit careful in the measurements, so you can just sort of in how you do the scaling. One thing I've been kind of interested in is, I think that type of experiment is difficult to interpret, but I think the one context where it could be interesting actually would be to do this at the level of single cell RNA sequencing. So maybe more interpretable in that context. So you could imagine that there may be variants that are not changing the mean level, but they're changing the fraction of cells that are expressing a particular gene at a substantial level. So we haven't done much in that direction. I think it's potentially interesting. Yes?
Presumably when one gets around to working in the transacting factors, we'll get at some of this issue
Of
variance in gene expression in specific genes.
potentially.
Some of the variance that you see in the expression of these cis-acting, in your cis-acting analysis, can presumably be attributed if one knew the transacting factor.
Yes, yes, I'm sure that's true. So that'll be variances on, presumably mostly uncorrelated with genotype at the cis locus, but
The question about the variance in the cis-acting expression, some of that is variance that's environmental, but a lot of it is going to be actually transacting.
agreed, yes. So Alcus Price has estimated that, I can't remember the number, I think maybe a bit more than half of the total variance in gene expression is due to transacting effects. I think that it's probably the case that most transacting effects start as a cis-acting change in one location in the genome that that then has transacting effects elsewhere. So I think it even though trans effects are responsible for a lot of the genetic variance, you can I think the key to understanding this still starts from cis. But I mean, I agree with your comment. Yeah, David.
Just following up on that, there was
Yeah.
Alkas's paper was really interesting in that there was two measurements of this. One of which was what proportion of the variance in gene expression was transacting. One of them was in blood, and the other was in cell culture, and there was a different proportion. And I think there was like a thought that the heterogeneity of the tissue type actually affected this. So if you're in a heterogeneous tissue type like blood, I think more will be transacting because it's because you're actually seeing many different tissue types and you're actually averaging over them. So the cis in one case it's not going to be acting, whereas cis in the other. So you actually see the things that are common across cells or the reverse.
Yeah, I'm wondering if it's the reverse. Is that possible? Yeah.
There's some issue like this, so it's hard to
Yeah.
measure as to partition the effects in terms of trends and cis because you're dealing with a certain population of cells that's a mixture of different types of activity.
Agreed. Yeah. So I think the one part of his result was that there's relatively little sharing of the trans heritability between the cell types, and so that's sort of consistent with the idea that you get reduced trans heritability in the mixed cell type. Yeah. I think it's a great paper actually.
Okay, so let's thank Jonathan once more.